Good day, everyone. My name is Irish Jody Constancio. Our topic for today is inductively coupled plasma spectrometry. Let's start in our topic. We have learning objectives. At the end of the lesson or session, students are expected to first explain why ICP, MS, or inductively coupled plasma mass spectrometry is particularly useful trace element analysis. Second is apply the process of ICP analysis and explore its applications. Third is express their findings and conclusion through a written or oral presentation. Next, introduction. Inductively coupled plasma spectrometry is an analytical method used to detect and measure elements to analyze chemical samples. The processing is based on the ionization of a samples by an extremely hot plasma, usually made from argon gas. The formation and characteristic of plasma when argon gas is heated above 6,000 Kelvin. At these high temperatures, argon transitions into a plasma state, which is characterized by the presence of a significant number of free electrons and ions. This plasma can be generated through methods such as DC arc discharge or inductive heating, specifically using an inductively coupled plasma or ICP torch. One notable method mentioned is the use of a Tesla coil to produce a high voltage discharge through flowing argon, which releases free electrons necessary for igniting the gas into a plasma state. Once the plasma is created and is confined within a high-frequency electromagnetic field, the resulting environment accelerates the ions and electrons, leading to increased collisions with both argon and any analytes present in the system. At temperature rising to around 10,000 Kelvin, energy transfer within the plasma becomes highly efficient, allowing it to sustain itself. The configuration of the plasma resembles a fireball, maintained in position by the surrounding magnetic field. This finding underscores the unique properties and behaviors of plasma in the context of high temperature and electromagnetic interactions. The process involving the sample aerosols and the resulting analysis techniques demonstrates the sophisticated interplay between plasma physics and analytical chemistry. As the aerosol sample enters the high-temperature fireball created by the plasma, it experiences rapid heating, causing the components to break down into atoms or ions, free from molecular associations. As these elements exist the plasma, they form a plume that contains the individual atoms or ions of the sample. As the plume cools to temperatures between 6,000 to 7,000 Kelvin, the excited atoms and ions transition to their ground states, leading to the emission of characteristic spectral lines. This phenomenon forms the basis of ICP atomic emission spectrometry or ICP-AES or ICP optical emission spectrometry or ICP-OE, which allows for qualitative and quantitative analysis of the sample's elemental composition. In addition to being analyzed via spectroscopy, a portion of the plume can be redirected into a mass spectrometer. This technique known enables the determination of isotopic masses for individual elements present in the sample. ICP-MS provides high sensitivity and precision, making it particularly valuable for trace element analysis. Both ICP atomic emission spectrometry and ICP-MS facilitate quantitative measurements, allowing researchers and analysts to determine the concentration of various elements in a sample with high accuracy. These techniques are widely employed in various fields including environmental monitoring, materials science, and food safety, among others. The operation of an inductively coupled plasma or ICP torch involves a carefully designated system that allows for efficient introduction of the sample while maintaining the integrity of plasma generation. In this setup, argon gas flows at a rate of 10 to 15 liters per minute through the three concentric quartz tubes of the torch, facilitating both plasma stabilization and sample transport. The outer tube directs and tangential flow of argon gas necessary for maintaining the plasma state. The argon gas movement contributes to the confinement and stability of the plasma, which is vital for effective heating and excitation of the sample. Within the central tube, nebulized sample droplets are introduced, suspended in argon. This configuration ensures that the sample is efficiently carried into the plasma without significant losses. The initiation of plasma occurs through a high-voltage ignition process, and it is sustained by a radiofrequency generator which typically provides 2 kilowatts of power at around 27. This combination of electrical energy and the magnetic field is critical for maintaining the high temperatures required for the operation of the inductively coupled plasma. Effective nebulization of the sample is also key to achieving optimal analysis. During this process, the sample is pumped into a nebulizer, which produces fine droplets that are easily carried into the plasma. Larger droplets do not achieve sufficient vaporization and are typically removed as waste from the spray chamber. It's recommended to avoid viscous solvent systems as they can hinder the nebulization process and lead to inconsistent results. Additionally, advancement in sample techniques have led to the development of high-solids nebulizers capable of introducing particulate matter and slurries directly into the ICP. This advancement enhances the versatility of the ICP for different types of samples. Other innovative methods such as laser ablation can also be employed, where a focused laser vaporizes the sample directly. This technique allows for direct analysis of solid samples and is particularly useful for materials that cannot be easily dissolved or aerosolized. Determining mercury in wastewater. ICP-MS or inductively coupled plasma mass spectrometry is particularly for analyzing mercury due to its ability to measure low concentration with high precision. The technique allows for the quantification of mercury levels essential for monitoring compliance with environmental regulation and assessing the impact of industrial discharge. In identifying lead isotopic composition naman po, yung lead or PB po, contamination can arise from various sources, including industrial emissions, leaded gasoline, and old plumbing. Different sources may result in varying isotopic composition of lead. ICP-MS facilitates the analysis of isotopes, helping to trace and identify the sources of environmental contamination through isotopic fingerprinting. Next, trace studies. ICP-MS can be employed in tracer studies where isotopes are used to track the movement and transformation of contaminants in the environment. This application is critical for understanding pollution pathways and informing remediation strategies. In chromatographic separation naman po, the technique can also be combined with chromatographic methods to separate different species of elements before mass spectrometric analysis. This is particularly useful for complex matrices where multiple species may interfere with measurements. By measuring a specific isotopes after separation, ICP-MS can enhance the specificity and accuracy of the analysis. And we have advantage of ICP-MS. Example, environmental analysis. High sensitivity. ICP-MS is renowned for its ability to detect trace levels of metals, typically down to parts per trillion concentrations, making it suitable for regulatory monitoring of hazardous substances like mercury and lead. And to rapid analysis, the technique allows for rapid multi-elements analysis, enabling the simultaneous detection of various contaminants in a single run, which is efficient compared to sequential method. Next is isotopic analysis. The capability to analyze isotopes adds a valuable dimension to the understanding of environmental contamination sources and processes, aiding in forensic and epidemiological investigation. And the multi-purpose applications beyond mercury and lead, ICP-MS can analyze a wide range of elements, making it versatile for different types of environmental samples, including soil, sediment, and biological materials. And interference naman po, the presence of a large excess of electrons in the ICP helps prevent significant ionization interference, which can be limiting factor in other methods. High temperature. In high temperature, the extremely high temperature, typically around 6,000 to 8,000 Kelvin, in the plasma effectively reduce interferences from molecular species and the sample matrix. This environment facilitates efficient excitation of elemental species and improves sensitivity and accuracy. And elemental emissions lines. ICP atomic emission spectrometry can simultaneously excite and detect a wide range of elemental emission lines. This allows the technique to analyze many elements in a single measurement, making it highly efficient. One example of the application of ICP are in environmental sciences. Students studying environmental chemistry can use ICP to analyze heavy metals in soils and water samples. For instance, a research project could focus on measuring the concentration of toxic elements like lead or mercury in local water bodies. By collecting water samples and analyzing them using ICP, students can gain insight into the pollution levels in their community, informing both local stakeholders and policymakers about potential environmental hazard. In terms of ICP-MS or inductively coupled plasma spectrometry or mass spectrometry represent a more advanced form of ICP that provides not only element composition but also isotopic information. An ideal real-life scenario for students would be its application in forensic science. For example, students could investigate a case that involves determining the source of a specific metal found at a crime scene. By employing ICP-MS to analyze trace elements from evidence like hair or soil samples, students can match the element signatures with potential sources, thus contributing valuable information for criminal investigations. This application underscores the interdisciplinary nature of science and demonstrates how advanced analytical techniques can support justice. In terms of atomic emission spectrometry or ICP-AES, often referred to, widely used in various industries including agriculture to analyze nutrient content in fertilizers. A practical application for students could involve conducting experiments to compare different fertilizer samples. By utilizing ICP-AES or atomic emission spectrometry to measure the concentrations of essential nutrients such as nitrogen, phosphorus, and potassium in various fertilizers, students can make educated decisions about which product may be most effective for their gardening or agricultural projects. These hands-on experiments not only reinforces classroom theory but also students with practical skill relevant to agricultural sciences.